going to take a little bit of time this morning to talk about what's next for SEPN1 and care. And so to begin things this morning, Hank Meyer is going to speak for a few minutes, and then we can feel free to ask questions along the way. Justin and I will be going around with the microphones, but Hank has a few words about what's next with SEPN1.
Okay, so I'm a pediatric pulmonologist, and I want to talk about, build on some of what we discussed yesterday. We spent a lot of the hour plus talking about respiratory insufficiency, sleep studies, and how we identify respiratory failure. And when we talk about respiratory failure, there are two different things that we look at. One is the inability to get enough oxygen, and two is the inability to get rid of enough carbon dioxide. Typically, when one gets sick, you have problems more with getting enough oxygen. With chronic respiratory failure, any neuromuscular condition, we worry much more about the inability of the body to get rid of carbon dioxide. And so, one of the things it's difficult to clearly identify hyperventilation with the precision needed using standard criteria. And that's been something that, for me as a non-sleep medicine person, has been very frustrating because I effectively interpret my sleep study interpretations from my sleep medicine colleagues. And so one of the things that occurred to me would be important is to better clarify how to how to precisely diagnose hyperventilation early, and not wait until I think the overly conservative criteria that the sleep medicine societies have for hyperventilation when that is met. And so one of the things that we did at our last pulmonary meeting about 2 months ago is I assembled a group of like-minded pulmonologists, many of whom had sleep medicine training, and some were adult practitioners, some were pediatric practitioners. And we started putting together the available criteria that we all use to define hyperventilation. And so the goal of doing this is to be able to ultimately create guidelines that can be disseminated that would allow somebody who's not necessarily experienced in managing patients with neuromuscular disease to be able to clearly and early identify hyperventilation. And the reason it's a bit of a challenge is that if you just look at the number, the upper limit of normal of carbon dioxide is 45 millimeters of mercury. The sleep medicine criteria say that you have to be above 50 for over a quarter of sleep. And the concern that I and many others have is what to do about that transition zone. And I feel that it's exceptionally important because in that zone you may be at risk of developing hyperventilation if something else changes. What could change? Well, you could get sick. And so one of the things that we want to do is to better identify people that are in that transition zone, and even one step above that, identifying patients who are in the zone where they're moving towards hyperventilation so that we can provide better guidance. And so I'm hoping that we'll be able to, we're right now going through a process of trying to identify overlap in opinion, and then we'll try to put some data to it and hopefully get criteria out within the next 12 months. So that's what I have. And in the next session, we'll be jumping into more management issues.
Great, thank you. And then I also wanted to invite Jaclyn up to say a few words. Jaclyn introduced a survey, a dietary survey yesterday, but it'd be good for us to hear just a little bit more because that's part of what's next.
So hi guys, hope everybody was able to make the presentation yesterday, looking at SELENON-related myopathy and the effect of diet and lifestyle on symptom severity in affected individuals. I know that there was flyers yesterday, so hopefully everyone was able to get one. And if you would like to sign up, like the contact information was on there. If you weren't able to find a flyer and are still interested or want to learn more, you can come find me, and I'll make sure that you get some more information.
Is that it? All right, thank you very much, Jaclyn. So, Lady and I have always felt like when we first got involved in this community, we always want to do something. So this survey is a great opportunity to do something to help impact the future of this work. So thank you so much. Before we get to Alan Beggs here, I have just one other announcement. On the end of each row, there is, in terms of doing something, Rachel had asked Lainey and I to put together a one-page document. It has to do with a fundraising event that we do every year, and we always tell these stories. So whenever we get to come to a conference like this, we share these stories with our friends and family. So if anybody's interested in doing event fundraising for the SEPN1 community, we're not experts, but we do one every year, and we're happy to help out in any way. And I think hopefully our slide should just about be ready. We'll give Alan the floor, and he'll talk a little bit more about what's next for SEPN1.
Sorry folks, we're almost there. Okay.
All right, and then we'll turn it over to Alan Beggs to talk more about what's next.
Okay, sorry, we're messing with the slides here. So, I thought I would just kind of briefly summarize some of the things that we talked about in the scientific session, in the 2 days before you guys all joined us. And then want to kind of tell you what we came up with for priorities and plans, but then ask for your guys' input and thoughts about priorities and plans. So, and I'll focus on the research side of things and progress and so on. In the next hour, we'll focus a lot on the clinical aspects of it. So clinical questions and management discussion will hold for that. So just one point I wanted to make is that we never try to put all our eggs in one basket. And there are a number of different approaches that are under consideration, not just in my lab, but in other labs around the world. We're all trying to think about this in terms of the common goal of establishing better treatments. Occasionally you may hear somebody say, call something a cure. I call that the C word. We try not to use the C word because it raises unrealistic expectations. But nevertheless, treatments can have tremendous benefit and impact on somebody's life. Yesterday you heard Anna Ferraro, Katie Mayer talk about antioxidant therapy. And in my mind, that's sort of like hitting a needle with a sledgehammer, because we believe that SELENON or SEPN1 has a very specific function in the cell, and having too much oxidation levels is part of that. But then when you take NAC, you're changing the entire cell, you're changing everything in the cell, not just the specific thing, in this case, perhaps the circuit channel, which we think might be one of the targets. So it may be useful, it's certainly safe, and appears to potentially have variable benefit in different people, but we want to do better than that. As you heard yesterday, Dr. Zito has identified other aspects of what's going wrong in the cells. So this endoplasmic reticulum stress, ER stress we call it, is one aspect of that, where proteins kind of get backed up in the machinery of making more proteins, and then it eventually can lead to the cells not working properly. And so there are drugs that are known about, or chemicals that can kind of impact that. So another approach, having understood the basic biology, is to kind of target that. And then circuit activators. We talked about, Bezad yesterday talked about the circuit channel, which is this protein responsible for taking calcium back up to essentially recharge the batteries for the next contraction. And so if we can identify chemicals, and there are, especially in the cardiology field, this is an important aspect of ensuring that the heart beats properly in heart failure. So there are pharmaceutical companies, for example, that are developing drugs to help keep the circuit channels active. And it may be that those have benefit in SELENON myopathy. And then finally, Dr. Ferraro and then our lab are doing a series of drug screens. And in a drug screen, you don't necessarily need to know what you're looking for, but you develop an assay. You basically, in our case, we're setting it up so we have normal cells that do one thing, and then we have SELENON deficient cells that do a different thing. Throw a bunch of chemicals on the deficient cells and see if whatever that aspect is can be corrected. And in some cases, Anna is looking at the levels of reactive oxygen species, the oxidation levels, or the effects of those chemicals on the bioenergetics of the cell. We're looking at the flow of calcium in the cell. And so there's really no way to know which of these approaches is better, but the thought is that we're going to each independently take our approach. We're going to tell each other what we find. If we find a chemical or a drug that seems to work in one of our assays, we'll tell the other person, and then they can test it in their system. I think this is really one of the nice things about this field is that the scientists and laboratories and clinicians all work cooperatively together in that way. And then finally, in my group, we're developing, or the early stages of developing what we hope might eventually be a gene replacement strategy. And as few as like 3 years ago, I wouldn't have thought to do this because there are too many hurdles and too many unknowns. But some of you probably know of the spinal muscular atrophy drug that was just approved, Zolgensma. And my research is also focused on myotubular myopathy, and there's a clinical trial now treating patients with that by the same method, by this AAV-based gene replacement strategy. And so with that progress, it gets easier to imagine that we can kind of jump some of the hurdles in this condition. Yesterday, Behzad talked a little bit about some of the technical aspects, essentially making the fully functional SELENON gene fit within the viral vector, finding a way to measure whether it's active and so on. These are difficult problems to some extent, but they're also surmountable. They're technical issues, and so it's just going to be a matter of time that we're working on. So, after the 2 days that we spent Thursday, Friday of this week in the scientific session, I came up with this slide to kind of suggest where we were and what we as physicians, scientists, and researchers were interested in doing. And so I'll go through this, but then I want to get you guys input. So I think one of the most important things, we can never do anything alone in either the clinic or in the academic research lab, or even no company can do anything alone. We really need to work together. I often talk about those three components as a triad. When I talk to companies and try to generate interest on their part to develop treatments for SELENON myopathy, the first question they ask invariably is, how many? And you know that we're one of the smaller groups here, but there's a lot more folks out there, we believe. And we have, we think we may be knowing about 1/10 to 1/5 of all the people on this planet walking around with SELENON myopathy. So the question is, how do we find more folks? How do we develop a robust community? Companies, when they're deciding to invest, whether or not to invest in a new therapy or treatment, they look to see what's the unmet medical need. Well, each of you as individuals clearly has an unmet medical need. That's something that we can demonstrate. But then they also want to know how many people is this going to impact. So, registry creation. I urge everyone to register with the CMDIR. Hopefully also enroll in my laboratory's research study. Casey Janetti talked about that yesterday. For those of you who already have a diagnosis, enrollment consists basically of going through it haven't finished my coffee yet at this point. Going through an informed consent process, having a chance to ask a lot of questions, and then essentially giving us permission to collect medical records, to stay in touch with you, to ask questions. In some cases, we may ask for a DNA sample, which will usually be in the form of a blood sample, but can often instead be in the form of a saliva sample. It's less necessary for people who already have a genetic diagnosis. For folks who've had a muscle biopsy, where that biopsy has been recent enough that it may still be in the hospital, we'll ask for permission to collect that muscle biopsy. And then we'll either study it ourselves, or in some cases we share it with folks like Dr. Zito or Dr. Ferraro. And then staying in touch. One thing is that we have 1,000 families in the laboratories registry, so we don't always reach out as often as we should, but we urge people to contact us, to send an email. If you're sending an email to me personally and I don't answer, send it again. Even better is send it to our research study coordinator, who right now is Casey, hopefully will be for a long time. Also, stay in close touch with your clinicians. Your clinicians are the folks who are most up to date on what's happening. And if you have a physician who is not understanding what you think some of the recent advances are or has questions, feel free to put them in touch with one of the folks that you're going to be hearing about in the next hour. And we all know each other. A lot of us, some of you know Kathy Matthews in Iowa. She's a good friend. She's collaborated in the past. She's actually been a co-author on a paper with Megan sitting in the back there and with us. It came out last year. And so we all work together. But put us in touch with each other. Actually, the other point I wanted to make is community building among you guys. And I believe you have a Facebook page, and specifically, I don't go on that. And not only do I not have a personal Facebook page for myself to simplify my life, but I also don't want to impinge on your guys' personal lives. But the utility of that page is for you guys to really have a robust community, talk amongst yourselves, and then reach out to us. Encourage other folks to join your Facebook group, encourage them to register, or at least contact us to get more information. The more we know about, the better. Other priorities are animal models. It's been frustrating that the biology of this particular gene protein is that it appears to be necessary at a certain stage during early development, but then it's kind of held in reserve until the cells get stressed, and then it kicks in. And so when we look at a zebrafish or a mouse or even a cell that's lacking SELENON, oftentimes it's very hard to tell what the abnormalities are. And so what we're learning now is that we have to stress them. So we need to understand that better, because if we don't have a sick mouse or a sick fish, we can't figure out if the drug or the treatment we're developing is going to be beneficial. And we really need to be able to show that before we can go to FDA or other regulators and suggest starting a clinical trial. Also importantly, again, coming back to it's the companies who really have the ability to translate our laboratory progress into clinical use. They're the people who can design and develop clinical trials. They have the experience with the regulators, the safety, and so on. And they're not going to be interested if we can't convince them that what we developed in a test tube is actually really going to work in a person. And then you just heard from Jackie a little bit about extra genetic factors. So those are some of the things that we thought were important. But I've got this slide. I want to sit down and type on it and try to get some feedback from you guys. I'm interested to know what you think are the most important things you'd like to see happen in the next 3 to 5 years. We can hope that maybe some of these things will happen faster. Probably some of these things may take longer, but it's really important to have a roadmap. So would anybody like to start and suggest what's the most important thing for you guys?
Raise your hand, I'll give you a mic.
As a mother, I would like to see you pursue what you talked about yesterday, an NAC test that's standardized.
So,
Why don't you
So
keep
in
that
other
one
words-
and rotate that one?
sure. So in other words, develop, when you say test, do you mean like a protocol for treatment? In other words, figure out what's the optimum for everybody to take, or how often and how much?
Or just choose a dosage and have everyone who wants to participate use that dosage and see what the results are.
Okay, that's a great idea. Since all caps, it didn't mean to, but that's okay. We're just gonna get it down. There are guidelines for NAC.
So I know like long-term goal is, you know, doing our best to treat it, but short-term is maybe just being able to get comfort in managing some of the symptoms like fatigue or decreased stamina. They aren't, you know, I think that maybe we could look also into more like treatments that might help, like maybe, I don't know, is swimming better for slowly building up stamina, or is walking something like maybe 10 minutes walk a day, would that be helpful in any way to kind of increase stamina?
Okay, that's great. So guidelines for management, and in particular you mentioned two things involving exercise.
Yeah, well, just decrease stamina and fatigue, those seem to be the biggest symptoms that I have to deal with day to day for my son-related food.
Right, and if you're going to go down this road, have something to track it with. Griffin wears a Fitbit, so he's tracking his resting heart rate, he's tracking his steps, he's tracking all sorts of things. So if you're going to go take the NAC and do the exercise, it's great to say it, but it's also be good feedback to have. What is your heart doing? Are you stressing your heart? These kind of things, because if not, we're just going to kind of be throwing darts at a wall again if we're not tracking what the body's reaction is to the NAC and exercise.
Yeah, that's a great idea. So there's a lot of development in terms of activity trackers and so on. There are also people that in informatics areas that are developing apps where you can just manage things. So certainly there are things like that for diet and calories and what you eat. You can do activity. And, so actually there is a department in my hospital called, it's an innovation department that basically utilizes computers and these types of informatics approaches to track things. Okay, those are great ideas. What else?
So I noticed, like for myself and I think for others, at a certain age, when you're older, maybe around 30s, it seems to kind of maybe get worse for some people. So I know it's hard, but maybe look into why.
Yeah.
Look what factors might affect that.
So that is, generally speaking, that's what we refer to as natural history. And actually Anna Ferrara has a pretty good handle on that in France. There's a paper that we've been working on, actually with some of you as subjects, with Francesco Montoni in London, to follow the natural history and track people over time. And I think that's critical. One of the things we know is that, for example, at a certain age, your respiratory capacity drops off, and that's when it's often important to go on things like BiPAP and potentially have sleep studies and so on. So knowing what to expect and when, and how the natural aging process affects you. So that, in our collecting records, is one way of doing it. It's called a cross-sectional study, where you look at a group of people at one point in time, but really what's important to do is what's called a longitudinal study, where we collect records today, and then we call you up a year from now, and then another year after that, and you can get more grandiose. You can have people come in and do pulmonary function tests and other tests. This is what Anna was talking about having done. Okay, that's what Anna was talking about having done in France for her NAC study. And she actually was a little bit over-ambitious, and it made the study difficult to complete for a large number of people. So the question is finding that sweet spot. But natural history studies, I'll put down here, is very important. The effect of aging.
I don't know if this is so much research, or it's more on the physician side, but a guideline of care for dietary management. Weight gain, how if too much weight gain is a bad thing for stamina and energy. Our daughter seems to maybe have some dysphagia, swallowing difficulties happening, chewing difficulties. That obviously plays into her dietary intake. So she's drinking a lot of her calories. So it's the long-term management of that. So I think that would be helpful, at least for us. I think there's probably others that are dealing with that as well.
Great, okay, actually Stephen just walked in the room, and in just a couple minutes we'll transition to the clinical management, and so that would be something that we should ask him about and incorporate into our future plans. Another question.
I think one of the major symptoms of SEPN1 has to do with scoliosis and orthopedic effects. And we have a technologist here, which is great, and can answer all our concerns with respect to lungs. But it would be nice to have an orthopedic person in these meetings, because it's definitely one of the major issues that we have to deal with on a day-to-day basis.
Absolutely, actually we were commenting that unfortunately we don't have an orthopedist on the panel today. Dr. Hollyardi and Dr. Day, sitting in the back, both have pretty extensive experience managing this. So we can ask them some questions, but I agree that we need to involve orthopedics more. And it may be that those specialists are seeing a number of community members who are, and not even realizing that it's SELENON myopathy, and certainly not integrating into this community. So